Hello students, in this video we are going to study about the very first kingdom which was given by Whitaker, that is Monera. As in earlier video we studied about the five kingdom system of classification, that is Monera, Protista, Fungi, Plantae and Animalia. So today we will discuss in detail about the Monera. So there are major groups involved in Monera, that are Eubacteria, second is Archibacteria, this is also pronounced as Archie, then Cyanobacteria, which is also called as blue green algae, then Mycoplasma and Actinomycetes. Actinomycetes, they look like fungus. They are also known as filamentous bacteria. Filamentous bacteria. So there are five groups: Eubacteria, Archibacteria, Cyanobacteria, Mycoplasma and Actinomycetes. So Eubacteria, it is also known as true bacteria. So there are some general features of Eubacteria which I am going to discuss here. First thing, you just see the diagram of this bacteria. This is a bacterial cell. You can see there is a outermost covering which is known as cell wall. Then there is plasma membrane. Then you can see there is no any cell organelles present in them, but there is some coiled structure which is the DNA, and it is coiled in the form of nucleoid. There is no structure which is called as nucleus. And outermost covering is the cell wall, and there is another membrane, or you can say covering present, which may or may not be present in all the bacteria, and that is known as capsule. So this is once in bacteria capsule is present, it is called as capsulated bacteria, and when it is not present, then it is called as non-capsulated bacteria. So you will study in detail in the chapters the cell, where we will discuss about this capsule, and there are so many layers like glycocalyx and slime layer. So here you see that this is flagellum, then full of cytoplasm, plasma membrane. There are some of course flagella like structure, but it's not flagella. Some hair like structure which is known as pili or pili, and then ribosomes. Here 70s type of ribosomes are present, and they are in the form of polyribosomes. Poly means many, and these are the cytoplasmic inclusions, and this is the uh, structure, some convoluted structure which emerges from the membrane, and this is known as mesosomes. So in this you can say the structure sometimes present and the structure always present. So the structure which is sometime present, it is flagellum, and there is some invagination of plasma membrane to form photosynthetic membrane. It is not drawn here. And this one, pili, it is not always present in all the bacteria. And the structure which is always present, that is the cell wall, that is the ribosomes, that is the cytoplasm, that is the DNA and plasma membrane, and also the food reserves which I drawn at cytoplasmic inclusions. Food reserves, it may be lipid glycogen or phosphate, etc. So these structures are always present in a bacterial cell. So let's talk about the general feature of bacteria. First thing, bacteria was discovered by Antony von Leeuwenhoek. I think you all know about him. In the year 1676. So Antony von Leeuwenhoek. And the term bacteria was coined by Ehrenberg. Term bacteria coined by Ehrenberg. So what are the general features of bacteria? First thing, they belong to kingdom-monera. Second thing, all monerans are unicellular. All are what? They all are unicellular. Next thing, they lack true nuclei. As you can see there, the DNA are arranged in the form of nucleoid, and other membrane bound cell organelles are absent. So what are the membrane bound cell organelles? It may be mitochondria, chloroplast, lysosomes, Golgi apparatus or Golgi body, etc. So these are the absent. Then they have rigid cell wall, and this is made up of peptidoglycan, which is very most important. This is also called as murin, murin, REIN. So cell wall is made up of peptidoglycan, which is also known as murin, and it is the polymeric unit of an acetyl muramic acid and an acetyl glutamic acid. So this in short is known as NAM, and this one is known as NAG. So an acetyl muramic acid and an acetyl glutamic acid. This is the uh, unit of this peptidoglycan, and this NAM and NAG, they are arranged like this. And detail of this we will study in the cell chapter. Next, the DNA has little or no protein associated with it. So DNA don't have any type of protein with it, and coiled DNA strand lie in the cytoplasm, so there is no any nuclear membrane present, so the structure is known as nucleoid, because it should not be um, distributed all over in the cytoplasm, so it is arranged in the form of nucleoid. Then flagella is if present, as I told you that it is not necessary that every bacteria has flagella, so if present it is a single thread and made up of several identical protein subunits called flagellin. Next thing, reproduction in the kingdom-monera is by binary fission, and they may undergo genetic recombination. This is, you can say, it is a type of parasexuality or a type of sexual reproduction in which they show genetic recombination. Next thing, they show extreme variation in their mode of nutrition. Most of them are heterotrophs. uh, and they absorb their nutrition in the form of solution, and some are also autotrophic, and it forms prepare their food by reducing CO2 and using either light energy or uh, autotrophs. When they use light energy, and once they use chemical, so once they use light energy they are called as photoautotrophs, and when they use energy derived from chemical reaction, they are called chemoautotrophs. And beside this, they also show symbiotic relationship, and I think you all have studied about the rhizobium, nostoc, anabina. So rhizobium is bacteria, nostoc and anabina is cyanobacteria, and some are also parasites. So they show diversity in their mode of nutrition. Some are heterotrophs, they may be parasitic or saprophytic. Autotrophs, they may derive their energy from light or from chemical reaction, and they may show symbiotic relationship, and there are so many examples of bacteria and cyanobacteria which shows the symbiotic relationship. So next, monerans do not have the ability of fixing nitrogen, but cyanobacteria can fix nitrogen. So we will discuss in next video how the cyanobacteria is able to fix nitrogen. And many monorails they can cause serious diseases in plants and human beings, and also they are the most important decomposers. And some other points like they I told you that their DNA does not have any protein, and that protein name is histone. So their DNA is double stranded circular DNA, but it lacks histone protein. It does not have histone protein. It is not bounded by histone protein. So shape of bacteria, there are many shapes of bacteria. It may be bacillus, spiral, comma, and bacillus, spiral, comma, coccus, etc. And they also comes in different sizes. So there are many sizes. The average size of bacterial cell is 0.5 to 1 micron meter. So there is another structure here. You can see here, this is mesosomes. Their function is not very accurate, but it seems that these are vesicular, lamellar or tubular pockets of membrane surrounded by the invagination of the plasma membrane, and it is most common in gram positive bacteria. And they have so many enzymes which can take part in respiration process. So I took another term, that is gram positive bacteria. So this you will also study in the chapter cell. What is the meaning of gram positive and gram negative bacteria? So this is all about the eubacteria.